koutou. Ko Kati Erekehu tōku hapu, ko Wairewa tōku marae, ko Kaitahu tōku iwi, ko Megan Leask ho. So today, we better go on to my slides. I wanted to talk about precision medicine and the genetic and molecular approaches that my lab are starting to take for this down in Dunedin. So currently in the health system, we have a one-size-fits-all, a kind of bog standard approach to medical care. If you guys go to the doctor for a checkup, you're likely going to spend 15 minutes with them. They might ask a couple of personal questions. They might regrettably put you on the scales to take your weight. And if they see from your blood sample that you have high cholesterol, they're likely going to put you on the same statin as every other patient that they prescribe. A precision medicine era is fast approaching and set to become routine in healthcare in the next decade. And so it's this tailoring of our disease prevention and treatment. It takes into account people's lifestyles, their environments, and more importantly, their genetics. Genetic information is just one way that this will come into place in the future. And I guess it's important to point out that over in the US, they are already using genetics to inform healthcare. Genetic scores are used to assess people's risk, and those in high-risk categories are receiving interventions earlier based on their genetic data and their environmental risk factors. What's important to know is that the genetic data that are used in these screens do not contain Māori or Pacific data. So a status quo precision medicine approach actually threatens to exacerbate the health inequities that we see in Aotearoa. And to prevent this, it's critical that genetic approaches to precision medicine are undertaken in Aotearoa for Aotearoa. So if we are going to start using these genetic scores in clinical practice in Aotearoa, we need lots of genetic data, and we need to know that the scores are actually going to work for Māori and Pacific peoples, and nobody's done this yet. There's plenty of literature out there that indicates that these scores that are derived from European people, for which we have the most genetic data, do not accurately predict disease for minority populations. But we don't know if this is true for Māori or Pacific peoples. In addition to this, the scores that have been generated from genetic data within populations and then applied to those same populations appear to fare a lot better than those that have been generated in Europeans. And so in my research, my first analyses with my team, we're going to answer these questions. Do European scores work for Māori? Do population-specific scores work for Māori? So we are testing whether or not, through collaborators, we have access to genome-wide data for Europeans. We've got 2 million Europeans genetic data that we have for gout. And then we have 1,000 people from Tahiti, so they're what we would consider people of East Polynesian descent. And we also have 3,000 people of Hawaiian ancestry that live in the US. We're going to try different combinations of data. We're going to combine all of it, so Tahiti, Hawaiian and European data, to see if ancestry or a pan-ancestry score works better than a European score or works better than a population-specific score. And then we will apply this to a data set of Māori and Pacific peoples that we have of about 2,500 people. And this is what we call the training set. And then from this, we then apply this to another set of Pacific and Māori data sets. We have a predominantly Māori data set from our community relationship with Ngāti Porou Oranga, and we have a unique Pacific data set as well from our community partners, the Pukapukan community. So I haven't done this yet. We're sort of step 2-ish. But the results of this approach, whether or not they are negative or positive, are important either way. If they show us that the scores generated in this way are successful and comparable to what we see for Europeans, then we can be confident that we can use them in clinic, but we can also use them to start identifying biological targets that might be targeted for therapeutics. If they don't work, this is important information for Aotearoa, and we can leverage this to identify new genetic strategies which we could apply for Māori and Pacific populations. So in parallel to this work, the other thing that I'm doing in my lab is to establish functional pipelines. When we find genetic variants or genetic discoveries, we need ways of testing whether or not they are good therapeutic targets, what tissues they are affecting, and what they're actually doing in terms of biology. One of my organisms that I work with are zebrafish, and you might be like, zebrafish, what? Why would we use those? But they're actually a wonderful model. They recapitulate human development quite nicely, and they're a hell of a lot more cheaper than our other organisms like mice and rats. And so what we can do is we can quickly assess what these genetic variants are doing in zebrafish, and then prioritise whether or not we want to take them through to more complex assays that we use in cell lines and maybe mice and rats. And so that leads me to the next thing we would be doing is understanding those biological and molecular mechanisms, maybe by knocking a gene out or knocking a gene in, because you really need to understand all those biological pathways if you're ever going to develop a therapeutic. And then there's this black box. And I think it's a black box to me and many other scientists working in molecular biology, is that at some point we hope that we will develop a therapeutic. However, I'm the first to say that I have no clue how to translate my biology into therapy. So that's when I need my collaborators and my colleagues to help me with this. And so I'm currently collaborating with young drug developers who know a lot more about this field of research than I do, so that we can focus on those gene targets that seem to have the most promise and could be targeted in clinic. But what we really need to make precision medicine a success in Aotearoa, well, first things first, we always need more data. One of the reasons our scores might fail to predict disease is the fact that we're generating them in a very small cohort of people. Remember back to those 2 million European people that we have data for, we know that those scores work for Europeans. To achieve this, we can certainly work with our established communities to recruit more Māori and Pacific peoples. And this is entirely plausible in the next 5 to 10 years. We know that genomic sequencing costs are going to decrease, and at some point cost won't be a hindrance. The bottleneck will instead be capacity and capability in Aotearoa. We need culturally competent researchers that our communities can trust with their genetic data. Even better, we need communities that have the capacity to carry out this research themselves. So my current focus is two-pronged. One, establish those methods I described in consultation with our communities, so that we can ensure that tikanga is upheld, given that we are working with genetic data, which is taonga. And two, we need to, well, I would like to establish tuakana-taina relationships with the next generation of Māori and Pacific researchers, data scientists, molecular biologists, so that when the time comes, they can return to their own communities and start doing the mahi, regain tino rangatiratanga over this taonga and their hauora. I don't think I've got enough time to just do that. Finally, funding is always good to have as well, and I'm hopeful investment into science will increase in the next decade or so, so that we can support these wonderful young researchers into successful research careers. And with that, I just want to thank the organisers for inviting me to speak, the funding bodies who believe in this research, and my wonderful team that are enthusiastic, dedicated, and remind me daily why I'm doing this mahi. Kia ora.